So this is part of Lab 5 where I want to talk about the math that's used to actually quantify the bacteria from a soil sample. So it's describing how to do the calculations from the information that you obtain from actually performing the lab. So there's a variety of things that you have to keep in mind. So just a quick review of what we did to set up the experiment. And so you had a soil sample that you had in phosphate buffered saline. And what you did is you transferred 1 milliliter of that soil sample to 9 milliliters of phosphate buffered saline. So that was our first dilution. And we labeled that as 10 to the minus 1. And what that really means is that was a 10-fold dilution. So 1 milliliter of soil, 9 milliliters of phosphate buffered saline. And so if you look at that ratio, it adds up to 10 total parts of 10 total parts, 1 part soil, 9 parts PBS. And so what we've done is effectively diluted this soil sample a total of 10 times. So we call that a 1 to 10 dilution, or we can use this exponent form, 10 to the minus 1, which is 1/10. Then we took that tube, our 1 to 10 tube, and we took 1 milliliter out of that and combined it with 9 milliliters of PBS. So we did another 1 to 10 dilution. So 10 times 10 is 100. So we did 2 consecutive 10-fold dilutions. And so this second tube we write as 1 to 100 or 10 to the minus 2. So those are equivalent to one another. And we continued this through what's called a serial dilution for another 3 tubes. And so each of these tubes has either a negative exponent that we can use, so 10 to the minus 1 or 10 to the minus 2, but probably more helpful is to actually understand what that represents. And so think of this 10. If you multiply 10 by itself 3 times, you're going to get 1,000. If you multiply 10 times 10, you get 100. If you multiply 10 5 times, you get 10,000. And so this is telling you how many times our original sample has been diluted. So, for example, if there were 1 million bacteria in this original sample, then because we diluted it 10 times, this tube is going to have 10 times less than the original. This one will have 100 times less. This one will have 1,000 times less, etc. And so we're making these series of dilution with the idea that we're going to take a sample from each of these and put them onto a plate. And so that's what this is representing here. This is the actual volume that we transferred. So we did not put a full milliliter onto the plate, but instead we took 0.1 milliliter or 1/10 of a milliliter, and we put that onto a plate. And then over time, you were able to see if there were colonies that were growing. And on some of the plates, it may have been so many colonies that they kind of grew together into a giant sheet, and there really weren't great colonies to speak of. And so what you're trying to find is you don't have to count colonies on every plate. And so what you want to ask yourself is which one plate should I count? And what you're trying to find is a plate that has somewhere between 30 and 300 colonies on it. So this one's pretty easy to count. It shows there are just 4 colonies. And remember what a colony represents. So a colony represents a single bacterial cell landed in that spot. And over time, that one cell divided over and over again until you could see it with your eye. And so each colony represents a single bacterial cell that landed on that plate at the time that you actually put this volume onto the plate. So in this plate, we can say there were 4 bacterial cells that landed on this plate. 4 is not between 30 and 300. So this plate we're not overly interested in. This one looks like it has a lot on it. It has colonies, but there's so many. This one is within that countable range. And so if you wanted to, you could actually look and count those. And so our example here, it has 32 colonies growing on it, which is indeed between 30 and 300. And so that's what you want to do first, is count the appropriate plate. And so now we can take all this information and use it to do our calculations. So that's what you've done in your experiment. So now what do we do with all these numbers that we have in front of us? So again, you need 3 pieces of information to do this calculation. So the first is the number of bacterial colonies on a specific plate. So in our example, there were 32 colonies. You also need to know, well, what is the dilution on that specific plate? So if we go back, this plate is our 10 to the minus 4, or probably more helpful, we can write that as 1 to 10,000 dilution. So it's our 1 to 10,000 plate. So that's our dilution factor for that specific plate that we counted that has 32 colonies on it. And then also how much volume of bacteria was actually put onto the plate. Now this can vary because you guys have those 2 ml pipettes, so we could have just as easily put, instead of 0.1 milliliter and spread around, we could have put 1 ml, we could have put 2 ml, we could have put 1.5 ml. And so we need to know the actual volume that we put on the plate. And so the volume that we spread on that specific plate was 0.1 milliliter, or 1/10 of a milliliter. And so when you have those bits of information, these are the steps that you're going to follow to actually perform your calculation. So again, you count the colonies. So we counted 32. Then, so I have 32 colonies. Then we're going to multiply that colony number by whatever the dilution factor is on the plate. And so the plate was 1 to 10,000. And so we're going to multiply by 10,000. And the last thing you're going to do, and this is sometimes why it confuses students, so I'll tell you why we're multiplying by 10. So the reason we're multiplying by 10 is because of this last bit of information. We spread 0.1 milliliter, or 1/10 of 1 milliliter. And so what we really want to figure out is how much bacteria would be present in 1 full milliliter. And so this schematic I just made, really what we're calculating is how much bacteria is in this 1/10. And so if we add up 10 of those, or multiply by 10, then we're going to get a number that's not a fraction of a milliliter, not 1/10 of a milliliter, but rather it's going to be how much bacteria is in 1 full milliliter. And that's a much more helpful number. An example I give is when you are calculating the price of gas, the number that is presented is how much the cost per gallon of gas. You don't want to know how much it is per half a gallon or 1/10 of a gallon or 1/4 of a gallon. And so having a whole milliliter in this instance is much more helpful to calculate a bacterial concentration. So we're going to take that number, 32, that was the number of bacterial colonies. Again, that's the appropriate number. It's between 30 and 300, so that's the right plate. Then we're going to multiply by the dilution factor. Again, the dilution factor of that specific plate we counted was 1 to 10,000, so we're multiplying by 10,000. And then we're multiplying by 10. This is the exact same thing as dividing by 0.1. And so whichever way works better in your head, you can do this with a calculator, you can do it in your head, but realize what that is going to get you is 3,200,000. And these units, I haven't used this term yet, but it's CFUs. That stands for colony forming units. So number of bacterial cells in 1 whole milliliter. So not in 1/10, not in half a milliliter, not in 5 milliliters, but in 1 milliliter. So that's 3.2 million CFUs per mil. And that is frequently written because some of these can be really large numbers. We can be into, this is in the millions, but it could be billions or even trillions of bacteria potentially. And so that can be a lot of zeros. So rather than write it in kind of just standard notation, we often use scientific notation. And so this number, 3,200,000 CFUs per mil, can be converted to scientific notation. And so this first number is always going to be some number between 1 and 9. It can be 9.9. And then this tells you how many zeros you're going to put after it. So obviously what we're doing is we're 3.2 times a million will get you 3.2 million, which is what this is. And so this is the number of decimal places. So if you count, the decimal place is where I have my arrow right here. You'd have to count over 6 places, which is what that exponent is. And so 3.2 times 10 to the 6 is the scientific notation value of 3,200,000. So this is the best way to write this. And again, don't forget your units. And so we want to know what this number represents. So colony forming units, or CFUs. And also we want to know in what volume. And so imagine if I just said 3.2 million, that's a good amount of bacteria, but we really need to know in what volume that represents. So this is in 1 milliliter. It's not in 1 gallon. It's not in 1 liter. It's not in, an entire swimming pool, which is hundreds of gallons. And so this number has a really important meaning because it tells us how much bacteria is present in a specific volume. And this is now our estimate of how much bacteria was present in our original soil sample. So we made dilutions that made it a lot easier to count. So we had to count 32, but then using these calculations, we can get this 3.2 million number. So that's what we did.